Hello everybody, thank you for attending to my presentation. Thank you Liang Feng for inviting me. Sorry I couldn't make it live today, so I pre-recorded my talk. If you have any questions, don't hesitate to send me an email, and I hope you will like my presentation and have a good conference. Thank you. Today I'll be talking about leaf and canopy gas exchange processes in tropical forests. So a little bit about me. I'm Julien LAMOUR. I currently work in Toulouse in the south of France with Jérôme Chave. This is my second postdoc. I've done a first one in Long Island with Sean Serbin and Alistair Rogers. I've done multiple field campaigns in the tropics, particularly in Panama, but also in Manaus and French Guiana. And both postdocs were supported by the NG Tropics project, which is led by the US Department of Energy and involve a lot of fieldwork and also modeling. And the main aim is to produce a model called FATE, which aim at better representing the tropical forests and vegetation in these areas. So today I will first present why it is important to correctly represent gas exchanges in these vegetation models, and also detail what I've done in the past on leaf gas exchange processes, and also how to scale leaf gas exchange processes at the canopy scale and at the ecosystem scale. So first, to predict the effect of climate change on forest, but also the effect of forest on climate change, our main tool is Earth system models. So in these models there are several components, and an important one is the vegetation, which can be represented by different models such as FATES. So you know that vegetation captures a significant part of the CO2 we emit. We think that without the greening effect of the CO2 and the increase in biomass worldwide of the vegetation, without this effect today we wouldn't be at 430 ppm but much more. And vegetation also controls precipitation, so of course being able to correctly represent the carbon cycles and the water cycle is key to predict future climates. In 2014, Friedlandstein and al. Made an experiment where they compared the predictions of different vegetation models. So on the figure in the left, each line is actually a vegetation model, and you can see that they predict a different future. Some of them predict that the forest will stay CO2 sink, while others predict that forest will start emitting CO2. So that's a very different future. And to be able to lower the sources of uncertainty, what we clearly need is to improve the knowledge in the tropics, because they represent half the global forest biomass, but they're also very important in the CO2 and the water cycle. And the IPCC report details some sources of uncertainty in those models. And for example, we clearly simplify the plant diversity. We know that there are several thousands of tree species in the Amazon, for example, but we usually only represent 1 or 2 average species. There are also some processes that we know are not really well represented, such as the effect of temperature and drought on vegetation. And so to represent the effect of the environment on the vegetation, you have to know that in these vegetation models, even if the aim is to predict the climate or the effect of climate at the global scale, we rely on leaf gas exchange models. So of course, the first thing we need is to correctly represent the effect of the environment on leaf gas exchange. And then we need to correctly parameterize these models so they represent the huge diversity of leaves worldwide, and for example, the effect of environment, season, phenology, and species on leaf photosynthetic traits. So I will present the work I've done on improving the representation of gas exchange processes in vegetation models such as FATES, and you'll see that the fundamental research which is done on photosynthesis is really important to be included in these models. So when we talk about leaf gas exchange processes, there are actually three components. So the first one is gas transport. So usually these are physical models of the diffusion of CO2 and water between the atmosphere and the leaf. You also have a very important component, which is stomatal conductance. Stomata can open and close dynamically in minutes. So the plant saves some water, but by saving water and limiting transpiration, it also limits the quantity of CO2 that can enter the leaf. And the last component is the photosynthesis itself, which predicts the rate of CO2 assimilation based on the CO2 concentration in the chloroplast and the quantity of light and the temperature. So these three components and models were developed in the early 80s at the same time as the instrument to measure photosynthesis. But what I want to show today is that it is still a very active area of research. So for example, for the gas transport, a very early model of gas transport is fixed law of diffusion. Actually, in the gas exchange models and in the apparatus, it's not exactly fixed law of diffusion which is used. And this is because when there is CO2 diffusion that goes, from the atmosphere within the leaf, at the same time there is a huge quantity of water molecules that are evaporated, and you need to represent the collision between these molecules of water and CO2 to really model the CO2 inside the leaf. So that is what Cameron and Farquhar published in 81. And actually, this model of transport was changed very recently in a Nature Plant paper in 2021, and this was to better represent the diffusion through the cuticle, because in the past model, the assumption was that all the diffusion was through open air, for example, within the stomata canal. So of course, changing the models has impacts on the measurements we do as physiologists and also as modelers. So we published a letter in New Phytologist which explained what could be the consequences of such a new model. So the second part I worked on was stomatal conductance. When I started my postdoc at Brookhaven National Lab, the context was these two papers from the Medellin team. They had demonstrated that it was possible to represent the effect of the environment on stomata using a simple equation that they derived using the theory of optimal water use. So they published a first paper in 2011 and a second paper in Nature Climate Change, where they gave the parameterization of this model for different plant functional types around the world. So as a consequence of the optimal water use hypothesis and of the mathematical choices they made to derive the model, the equation predicts a linear relationship between stomatal conductance and carbon assimilation. And actually, the slope of this relationship is the optimal stomatal parameter, which is linked to the water use efficiency. And they had published several values for different kind of species worldwide. So when I started my postdoc, the idea was to go in the field and actually measure this parameter for different species of tropical trees, but also to see the effect of the phenological stage of the leaf on this parameter. But what we observed when we made the measurements is that instead of measuring a line, we measured a non-linear line. So at first we thought we had an issue with our protocol, but we repeated the measurements in different conditions, and we also used previous measurements made by another team, by Andrew Leakey's team, and we observed that this nonlinearity was present in all the measurements. So our interpretation is that stomata respond non-optimally at low light level, but that possibly the mathematical simplifications that are made in the Medellin model explain the nonlinearity at high light. So of course, when you change the model, it has impact on the simulation of the water and carbon cycle. So here on both figures, what you see is the effect of simulating transpiration and carbon assimilation when the light changes at the leaf surface. Of course, when the light changes, the temperature changes too, and you can see that you have drastic differences, which means that we still need to do empirical measurements and also test these models, everywhere and in different conditions. Most vegetation models represent the effect of the environment on photosynthesis using the equations from Francois, Van Kamera, and Berry developed in the 80s. So this model was slightly improved through times, and you can see a nice review by Xinyu Yin. In this model, the effect of light, particularly on electron transport rate, is modeled empirically. You've probably seen these two equations. So the second one is the solution from the first one, and they describe the effect of light on electron transport rate. So actually there are two states in this equation. The first one is a linear increase of electron transport rate with light, and the second one is actually the maximum value of electron transport rate, Jmax. And what does the equation do? Is actually smoothing the transition between the first state and the second state using a curvature factor, which is theta. So the lowest theta, the smoothest the transition between those two states. In most vegetation models, theta is fixed at either 0.7 or 0.9. Very recently, Johnson and Berry in 2021 proposed a new model for the effect of light on electron transport rate. So this is the same Joe Berry as in the early publication, but 40 years later. And what they did is actually propose a more mechanistic description of the effect of light on the electron transport system by describing the role of the cytochrome in the control of electron transport rate. So there are also another advantage of this model is that it predicts gas exchange but also fluorescence. So we were interested in comparing the two models, and the first thing we saw is that actually the second one from Johnson and Berry is analogous, at least the equation, because conceptually it was derived differently, but the equation is analogous with theta equals 0. So this was a surprise for us because, as I said, most vegetation models use a value between 0.7 and 0.9. So Johnson and Berry prediction models lower photosynthetic rates at intermediate irradiance, and this could have drastic consequences in vegetation models. So we have decided to compare the two models using more than 1000 photosynthesis slide curves to see if using a theta value of 0 was correct. And actually what we saw is that the Johnson and Berry model achieved better fits in most of the cases, and this was significant for all the biomes that we tested, so crop, gymnosperms, and temperate species, for example. So this paper it is really interesting. You should really read it. They gave a new look at the electron transport rate. We showed that it's a better fit in most cases, and even if we don't use these new equations, it can help simplify the parameterization of the Francois model using theta equals 0. And we have just submitted a paper really on discussing this new model and comparing the performances. To conclude this first part, you've seen that it's still a very active area of research. There are also many other works on leaf gas exchange processes, particularly on the mesophyll conductance, and also the effect of temperature on photosynthesis. And I think it's interesting to see that a strong assumption made in gas exchange theory is that the Humidity inside the leaf is 100%, but more and more studies show that it could not be the case, for example, at high temperature. Anyway, this very nice study from Noah and al. That was published in Science Advances, which shows that when you improve the representation of leaf scale processes, you improve the representation of gross primary productivity worldwide. So I really encourage you to read this paper. Now, when we measure leaf gas exchange traits at the leaf level, as I said in the beginning, a strong challenge is then to scale the traits and represent the leaf trait variation in tropical forests. I have been a co-author in different studies with Ken Davidson, who was a PhD student at the time, but also Angie Burnett, who studied the effect of phenology, light environment, and species differences on leaf photosynthetic traits. So you can go and see these papers if you're interested. For this kind of studies, we always rely on gas exchange measurements and then model fitting to derive the photosynthetic capacity or stomatal traits. A question you may have after my presentation is that what happened when we change the model? And actually that's why publishing the raw gas exchange data is so important, because if you published this raw data, when the model change, we can re-estimate the parameters, and of course this is important for science. Of course, when we change the model, we change the numerical values associated with leaf traits, but we can also, in some occasions, change our interpretation of ecological strategies. So here I wanted to show this study from 2023, where we went in the forest in Manaus, and we wanted to compare the stomatal control of gas exchange in early successional species and in late successional species. And on the same data, when you use a Medlin model of stomatal conductance or our model, you obtain a different conclusion. So with the first model, you will interpret that early successional species have a different stomatal control of gas exchange than late successional species. But with our model, you conclude that there are no differences. So of course, it's really important to use up-to-date and improved representation of gas exchange processes so we can really understand ecological strategies. So as the last message I wanted to tell in this presentation, so as you probably know, measuring gas exchange in tropical forest for ecological studies is tedious because it takes a long time, sometimes several hours just for one leaf. So this is a real limit to our studies. And very recently, it's been shown that using spectroscopy, so the instrument on the right, you can derive many leaf trait parameters. We have shown in 2021 that you can predict photosynthetic parameters using spectroscopy. So for example, the parameter Vcmax, but also Jmax and the dark respiration. Very recently, Fengqi Wu and Dengbing Yan have shown that you can also predict dark respiration even across forest types. And we have started this collaborative project with Sean Serbin and Alistair Rogers that I'm leading. And we are trying to combine as many datasets as possible that were measured in the past with paired observation of gas exchange and spectra. And so far we have more than 300 species, more than 4,000 spectra trait observations. So if you want to participate, if you want to see what we've done, here you can scan this QR code that will send you to our GitHub repository. If you have any questions, don't hesitate to send me an email. I'm always happy to talk about science and start new collaborations, so don't hesitate to reach out. Thank you very much.